Right. So, welcome back. So, now I'll discuss about classification of bioreactors based on mode of operation. Mode of operation means how we are going to operate these machines. In the earlier class, we had a look at what is a bioreactor and how we can classify based on size, right? So, here, this session we are going to discuss about mode of operations, right? How we can classify the bioreactors based on how we are going to operate the same machine, right? So we have three major modes of operating the bioreactor. First one is called batch mode, which is one of the most widely used mode till now. And second one, a fed batch mode, which is most widely used for producing Baker's yeast, antibiotics, and other biomolecules. And the third mode is called continuous culturing mode or continuous mode. So continuous mode is also used for producing products like vitamin C, propionic acid, as well as corn production. So we'll go one by one. This session I'm going to discuss about batch system, how we are going to operate a bioreactor in a batch mode. So this is the schematic representation of a bioreactor operating in a batch mode. So you have to remember one point, batch mode is called as a closed system because we are going to take our substrate, we are going to take our carbon source, nitrogen source, minerals, vitamins, whatever required for the growth of the organism, and inoculate the culture inside the reactor and then seal the entire system or the bioreactor. We are going to close it. And once we close it, we are not going to open the bioreactor unless and until the product is formed inside this bioreactor. So there is very minimal chance of contamination inside the batch system. That's why this is one of the most preferred method of producing biological products. So here, if you have a look at the schematic representation, we have the central vessel and which is connected by an air flow. Only thing that is going in and out of the bioreactor is the sterile air. Sterile air will be sent into the bioreactor during the process or during the course of bioconversion or bioreactions, and the air will be sent out of the bioreactor, which is nothing but released gases which are released by the microorganisms, which are unwanted gases which are released will be sent out of the bioreactor. So if you have a look at this picture carefully, so wherever something going in and something going out, there is some kind of a filter set for making the air sterile. Only sterile air is sent in, as well as the gases that are coming out also will be sterilized, and then they will be sent to a gas analyzer. If there are any poisonous gases, that will not be sent into that should not be sent into the atmosphere directly. That's why we should have a gas analyzer to analyze what type of gases that are going out of the reactor. Then only they will be sent out of the bioreactor, right? So as I was discussing, mode of operation of fed batch is very simple process. We are going to take a media which is going to use for production, sterilize it and put it inside the bioreactor, and then we are going to take the culture which has the ability to digest that substrate and produce the product. We are going to inoculate such kind of culture into the vessel, and then we are going to maintain all the environmental conditions which are suitable for that particular organisms to grow and then produce the product inside the bioreactor, right? So let's have a look at the advantages and disadvantages of the batch culturing system. So batch culturing system, as I said, which is one of the simplest type of culturing system, where we need to take the substrate and culture, put it in, close the system, and this is one of the easiest one. We can produce the product in as short as 24 to 48 hours of time. So maximum runtime for a batch system can go around something around 1 week. So within that time period, we can produce products inside a batch system. And second, a major advantage is operability and reliability. You can rely on the batch system because we can easily operate them, and we can, if you know the organism, if you know the ability of the organisms to produce the product, and if you know the what type of substrate we are giving, if you pre-plan all these things, we can easily get the product inside the batch systems. So we can rely on these machines for producing the product. Nothing will go wrong. And the third one is the production of secondary metabolites, right? So secondary metabolites, as we all know, are generally produced using the stationary phase, right? So if you want an organism to produce secondary metabolite, we need to make these organisms go into the stationary phase, right? So in case of batch system, there is a limited amount of substrate. Only once we are giving, we are not further giving any food materials to the organism. So whatever the food that is there inside the reactor will be consumed by the organisms. When the food gets over, then it has to go into the stationary phase, and during the stationary phase, they're going to produce these products. That is nothing but the secondary metabolites. So if you want to produce secondary metabolites like alkaloids or antibiotics or any other secondary metabolites, the ideal culturing system is nothing but your batch production system, because all the organisms will go into the stationary phase once the food's substrates get depleted inside the bioreactor. The next advantage is fewer possibilities of contamination. Since it's a closed system, we are not transferring anything into the reactor and taking out anything from the reactor. There is no manual intervention, so there is no chance of contamination. That's one major advantage. And the last one, unique batch number. So we can tag a unique batch number to the product that is produced from a bioreactor is a batch system. So that's one major advantage. Why? Because most of the biopharmaceutical drugs need to be labeled or tagged by a batch number. Without this batch number, the FDA and other regulating authorities will not accept any biopharmaceutical drugs. So batch number is essential, which can be labeled with a batch culturing system. So coming to disadvantages or the drawbacks, some of the drawbacks of batch culturing system. First one is culture aging and differentiation. So we have seen there is a limited amount of substrate inside the batch system. So once that substrate is completely utilized by the organisms will eventually move to stationary phase and into the death phase. So organisms will age, will get differentiated, and they will die. They undergo autolysis inside the bioreactor. So we cannot expect large amount of products because of limited time span and limited substrate availability inside the bioreactor. One drawback. Second one, buildup of toxic metabolites. So during these growth phases, when they are reaching the stationary phase and death phase, several toxic metabolites are synthesized by these organisms. They will be released into the broth, or when the cells undergo autolysis, this will be released again into the broth, spread inside the broth, and make the downstream processing of our product difficult. That's one major drawback of this batch system. Third one, initial substrate concentrations. We cannot put too much amount of substrate into the batch system. What happens is, if you give large amount of substrate to the batch system, the initial higher substrate concentration act as a limiting factor or inhibition factor for the growth of the cell. It will have a repressive effect on growth of the cells as well as the product formation. This is another major point which we need to remember. High substrate concentrations in case of batch system have a repressive effect on growth as well as product formation in the batch system. That is one major concern or a drawback. Next one, batch to batch variability. Even though exactly we'll do the batch operation, there is some minor differences in the productivity or the quality of the product from one batch to another batch. This is always there in the batch system. Increased non-productive period. So there is always a gap period between one batch and another batch production. Why? Because once the batch production is done, we need to remove all the unwanted material from the reactor, clean the reactor, sterilize the reactor, and there is a downtime for doing all these purposes, right? So that's why there is always a non-productive period involved with batch system. Next drawback is organisms from one bioprocess cannot be seeded into another reactor. So that's another major drawback. So the culture which is present in the current bioreactor cannot be transferred into another bioreactor. Why? Because the culture will get aged in this bioreactor and they get even mutated at the end of the bioprocess. So we cannot expect the culture to grow in the fresh bioreactor and produce the product exactly the same as how it has done in the earlier reactor. Okay, so that's one major drawback. We cannot transfer the culture from the one bioreactor to another bioreactor in case of batch mode of operation. So as I said, another major drawback is cellular autolysis, which will harden our downstream processing of the product during the course of time. So this is about batch system and major advantages and drawbacks. I'll wind up here. I'll continue with the fed batch system in the next session. Thank you.